FluDETECT Avian Influenza Virus Type A Antigen Test is an in vitro rapid immunomigration (RIM) test designed to aid in the qualitative detection of avian influenza virus type A in tracheal, oropharyngeal, or cloacal samples from symptomatic birds or flocks. This assay detects all 16 subtypes of influenza type A virus. Positive results should be submitted to a reference lab for confirmation and subtype determination. Negative results indicate that no detectable avian influenza type A virus is present. FluDETECT AIV is highly specific, sensitive, simple, and can be performed at the farm or in the laboratory. Test results can be obtained in approximately 15 minutes. The following items are packaged in each FluDETECT AIV test kit. 1 vial of 20 test strips. 1 6 milliliter dropper bottle of extraction buffer. 20 swabs. 20 test tubes, 20 test tube caps, 1 test tube rack for 5 test tubes, and 1 directional insert. Be sure to examine the test kit contents before testing begins. Sample extractions may be performed using either of 2 methods. If you use the extraction buffer method, the extraction buffer is provided for you in the FluDETECT AIV test kit. First, place 8 drops, approximately 250 microliters of extraction buffer in the test tube provided. Next, place the swab containing the specimen in the tube and rotate the swab 5 to 10 times in the buffer. When removing the swab from the tube, press it against the side of the tube repeatedly until no more liquid comes out of the swab. Discard the swab in an appropriate biohazard container. If the extracted sample will not be tested immediately, cap the tube with one of the caps provided in your kit and store the sample. The second extraction method is using brain heart infusion or BHI broth, which is not provided. Please also see your directional insert for a list of additional viral transport media alternatives. To begin the BHI broth process, first place approximately 500 microliters of BHI broth into the test collection tube. Next, place the swab containing the specimen in the tube and rotate the swab 5 to 10 times in the broth. When removing the swab from the tube, press it against the side of the tube repeatedly until no more liquid comes out of the swab. Discard the swab in an appropriate biohazard container. If the extracted sample will not be tested immediately, cap the tube with one of the caps provided in your kit and store the sample. Note, for the BHI extraction method, as many as 5 swab samples can be pooled into 2.5 milliliters of BHI broth. If samples are not to be tested immediately, they should be stored in the appropriate manner. Samples can be stored for up to 48 hours at 4 degrees Celsius or 39 degrees Fahrenheit. For more prolonged storage, samples should be kept frozen at -70 degrees Celsius. Do not store samples at -20 degrees Celsius. Do not store samples in a self-defrosting freezer and avoid multiple freeze-thaw cycles. Test kits should be stored between 2 degrees and 30 degrees Celsius, 35 degrees to 86 degrees Fahrenheit. Do not freeze test kits. There are 2 methods for testing samples. The method you use will depend on the sample extraction method that was used in the previous step. Be sure to follow the proper test procedure that matches the sample preparation method used. To begin, remove a test strip from the desiccant vial for each sample to be tested. Handle the test strip on the labeled portion of the strip. Please note, prior to use, the test and control bands appear yellow. The bands are dyed yellow for quality control purposes. The dye does not interfere with the test results and will wash away while the test is developing. Place the test strips directly into the test tube containing the sample. Position the test strip so that the pink pad is submerged in the extracted sample. Allow the strip to incubate in the sample for 15 minutes. Remove the test strip from the test tube to read the results. If sample extraction was performed using BHI broth or another viral transport media, use the following method to test samples. First, place 200 microliters of the viral transport media containing the extracted sample into the test tube provided. Next, add 3 drops of extraction buffer to the test tube. Tap the side of the tube to mix. Then, remove a test strip from the desiccant vial for each sample to be tested. Handle the strip on the labeled portion only. Place the test strips directly into the test tube containing the sample. Position the test strip so that the pink pad is submerged in the extracted sample. Allow the strip to incubate in the sample for 15 minutes. Remove the test strip from the test tube to read results. After 15 minutes, examine the test strip for the presence or absence of pink-purple bands in the center of the test strip between the two absorption pads. The control band appears in the upper end of the test strip, closest to the handle, while the sample test results are read in the lower portion of the strip. After reading the results, discard the test strip in an appropriate biohazard container. The test is valid if the control line, pink-purple band, develops in the upper part of the test strip. The absence of the control line indicates that the test is invalid and must be repeated. A positive result for avian influenza type A virus is indicated when two pink-purple bands, a control line and a test line, are clearly visible on the test strip. This result indicates that detectable levels of influenza type A virus are present in the sample. There are a few precautions to be aware of before using FluDETECT AIV. Do not use the kit components past their expiration date. Allow samples to come to room temperature, 15 to 30 degrees Celsius or 59 to 86 degrees Fahrenheit, prior to use. The vial holding the test strips contains a desiccant and should be kept tightly closed when not in use. Use the test strips within 10 minutes of removal from the desiccant vial. The test strip should only be handled in the upper, labeled region. Avoid contact with the surface of the test strip. The test strip should be placed in the test tube vertically. Use a separate swab for each sample. Swabs with wooden handles or swabs containing calcium alginate may interfere with the test and must not be used. Do not centrifuge samples prior to use. Swabs containing visible blood may partly obscure a weak positive band due to hemoglobin background or discoloration. Sample extractions of cloacal swabs containing an excess of fecal material may interfere with the test. Allow the feces to settle to the bottom of the tube, then draw the sample off from the top for testing. Remember, positive samples must be submitted to an approved reference laboratory for confirmation and subtype determination. A negative result for avian influenza A virus is indicated when a single pink-purple band or control line is present in the upper portion of the test strip. This result indicates that no detectable avian influenza type A virus is present in the sample. Very faint lines may be due to nonspecific binding and should be further investigated. Please note, the control line on the upper portion of the test strip may appear earlier. This does not mean the test is complete. The test strip must incubate for a full 15 minutes before the sample is interpreted as negative. However, the test can be considered to be complete if the test line on the lower portion of the strip appears before the 15-minute incubation period is over. This sample is interpreted as positive. If the test strip remains in the test tube for more than 20 minutes, a false positive ghost band could appear in the place of the reaction band. This video provides a general overview of the procedure for performing the FluDETECT AIV test. For details, please read the directional insert that accompanies each test kit and follow the detailed description of the protocol in agreement with good laboratory practices. For assistance, please contact Zoetis at 1-888-ZOETIS-1 or 1-888-963-8471.